everybody to this webinar organized by the European Reference Network for Neuromuscular Disorders, EURO-NMD, in collaboration with the European Reference Network for Rare Neurological Disorders and the European Academy of Neurology. And today we are going to have the pleasure of listening to Montserrat Olivé Plana. Montserrat is currently the director of the Neuromuscular Disorders Unit at the Neurology Service of the Hospital Santa Creu e São Paulo in Barcelona. And she has a training much centered on neuromuscular diseases, with a differentiation both in clinical neurophysiology, that I ignored, and neuropathology. She has an extensive number of publications, is a member of the research committee of the European Neuromuscular Center, the ENMC, and the co-chair of the Protein Aggregate Myopathies Consortium at the European Neuromuscular Center. She's a member of the Muscle Pathology Working Group of the European Network for Neuromuscular Disorders, the EURO-NMD, member of the Executive Committee of the same network, and co-author of the document Recommended Standards for Muscle Pathology. She's written 2 chapters of the book Muscle Diseases, Pathology and Genetics that was published under the International Society of Neuropathology. And she's a regular lecturer at the Summer School of Myology, organized in Paris by the Institute of Myology, as well as the Latin American Summer School of Myology. So after a long and distinguished introduction, Montse, I have the pleasure to give the floor to you. Thank you.
Okay, thank you very much, Antonio, for this kind introduction and presentation. Well, in this webinar, I will try to summarize or to give an overview of the myopathology of vacuolar myopathies and protein aggregate myopathies. And you can imagine that the number of conditions that belong to this group of disorders is huge. And there is an overlap with some pathological features among some of the disorders included here. So I will not speak about all of them. Of course, I will try to, as I mentioned, give an overview. I have no disclosures to declare, and this is the thread of my talk. I will start with a question to the audience and a brief introduction. Then I will go through protein aggregate myopathies with and without vacuoles. And then in the second part of my talk, I will speak about vacuolar myopathies, including rim vacuolar myopathies, autophagic vacuolar myopathies, and other vacuolar myopathies. And as I mentioned, I would like to start with a question to the audience. And the question is, in the area of massive gene sequencing, do we still need a muscle biopsy? And well, I cannot see your faces. I don't know if you agree or you disagree. We could make a kind of votation, or at the end of my talk, I can have your answers if you want. Anyway, my personal opinion is that a muscle biopsy is not always needed, but it still can be very useful in many situations. Muscle biopsy is needed for the diagnosis of not genetic conditions that may mimic an hereditary myopathy. Muscle biopsy helps us to differentiate a neurogenic disorder from a myopathy, in some cases in which results of the EMG exam are confusing. Muscle biopsy plays a major role in investigating the relevance or the significance of genetic variants of an uncertain significance. If we come across a variant in a new gene, muscle biopsy provides us important information to explain the pathogenesis of the disease. And finally, muscle biopsy provides us samples for research purposes. Well, I will start with protein aggregate myopathies, and as the name implies, these are disorders that are morphologically defined by the presence of protein aggregates in muscle cells. We don't have a good classification, I would say, to classify protein aggregate myopathies. And for practical purposes, I have divided them into hereditary and non-hereditary. And among the hereditary conditions, these disorders usually manifest as congenital myopathies or in early infancy or in early adulthood. And in the majority of cases, or in most of the cases, these disorders results from defective protein synthesis. And in these situations, the protein aggregates are not found in combination with vacuoles. There are no vacuoles. In contrast, protein aggregate myopathies manifesting later in life and usually result from defects on protein degradation systems in the muscle cells. And in these situations, protein aggregates are often found in combination with rim vacuoles. Among the non-hereditary conditions, inclusion body myositis is probably the best example. Well, why do proteins aggregate? They can aggregate because there is a defective synthesis, or because the proteins are misfolded, or because there is a defective degradation, or a combination of this phenomenon. Well, you know very well that protein homeostasis is essential for the normal functioning of cells and tissues. Chaperones help to properly fold native proteins and native polypeptides. These chaperones, as I mentioned, helps the proteins to fold, or can unfold and refold misfolded proteins. Some of these misfolded proteins, if necessary, are targeted to be degraded by the ubiquitin proteasome system or by the chaperone-mediated autophagy. But if these systems fail, then these misfolded proteins form aggregates, and these aggregates are targeted to be degraded by macroautophagy within the lysosomes. I will start showing you some protein aggregate myopathies with vacuoles, and I will not go through all these conditions. I will show the disorders that are written in bold letters. And the first one is myosin storage myopathy. Well, this is an autosomal dominant and less frequently autosomal recessive condition that results from mutations in MYH7, a gene encoding the slow beta cardiac myosin. The clinical manifestation of this disease vary with an onset that can be during childhood or young adults, and usually manifest with weakness in proximal muscles of forelimbs, or in scapuloperoneal muscles, or maybe sometimes including distal muscles. Cardiomyopathy is found in a fraction of patients, and the muscle biopsy typically show these subsarcolemmal hyaline masses that are observed in several fibers. This one is very well observed here. These hyaline masses correspond to slow myosin, as revealed with antibodies that recognize slow myosin. The muscle fibers, or the areas of the muscle fibers that contain these hyaline bodies or hyaline inclusions, are devoid of oxidative enzyme activity and do not contain proteins that belong to the Z-disc. Under the electron microscopy, the hyaline material corresponds to partially granular and filamentous material, which is not limited by a membrane. the pathogenesis of this disorder is supposed to be defective myosin filament assembly. And the mutations that cause this disorder are usually located in this end of the tail of the slow beta cardiac myosin, corresponding to the 37 to 40 exons of the gene. Well, some years ago, we described a new disorder that we call morph myopathy, which is very similar from the pathological point of view to myosin storage myopathy. And we identified this disease in this patient that manifested during adolescence with an hypertrophic cardiomyopathy, and he later developed proximal and distal weakness in the forelimbs and paraspinal muscles. He also had a rigidity of spine, mild scapular winging, and prominent muscle hypertrophy with almost lack of subcutaneous fatty tissue. The muscle biopsy of this patient shows subsarcolemmal inclusions that contain slow myosin, as we have just seen in myosin storage myopathy, and also contain other myosin-associated proteins, but were devoid of desmin, myothelin, and other Z-disc-associated proteins. In contrast to myosin slow storage myopathy, under the electron microscopy, the lesions observed in this patient were very particular because they corresponded to fragmented sarcomeres that were that have a preserved A bands and M line, but were completely absent Z lines, as you can see here. Well, interestingly, and in collaboration with Omar Daghsarhi and Anders Olfors from Gothenburg, we came across this paper that was describing a double knockout mice for MARF1 and MARF3, which are ubiquitin ligases. And these mice show subsarcolemmal accumulation of slow myosin and the same ultrastructural features as observed in our patient. So Oma was able to identify a mutation, an homozygous mutation in MARF1 in our patient in combination with heterogeneous mutation in MARF3. So this disease results from a degenic disease caused by mutations in genes that encode ubiquitin ligases that participate in sarcomeric protein degradation. Okay, let's move to reducing body myopathy, a disease that you can find it classified as myofibrillar myopathy because the pathological features, but also as a congenital myopathy sometimes. Well, anyway, this is a disorder that results from mutation in FHL1. It's a disease that is transmitted following an X-linked inheritance that manifests in infancy or childhood, very rarely in adulthood. Female carriers can be symptomatic, and affected patients usually present delayed motor milestones, proximal, axial, and limb weakness, rigidity of spine, scoliosis, respiratory failure, and the disease is usually fatal with early death, usually before 10 years of age. And the pathology of this disorder is very peculiar for the presence of the reducing bodies that are these bring bright pink inclusions observed in hematoxylin and eosin stains. These are the reducing bodies that appear red or dark blue with a modified trichrome stain. These reducing bodies are devoid of oxidative enzyme activity as revealed with SDH, but react with nitroblue tetrazolium menadione reaction. As I mentioned, and this is the cause, or this is the reason why the disease is called reducing body myopathy. While these images correspond to some cases I was I had the opportunity to study when I was working in Paris at the Institute of Myology with Norma Romero some years ago. Well, anyway, the genetic cause of the disease was discovered after the identification of FHL1 as the major protein component of the reducing bodies. And in addition to FHL1, muscle fibers contain also accumulations of desmin that are usually seen not within the reducing bodies, but at the periphery of them. If we look under the electron microscopy, the reducing bodies originate from material that has an appearance of coarse tubulofilaments that initially emanate at the level of Z lines and spreads through the sarcomeres and also under the sarcolemma. In this disease, there is typically a combination of this coarse tubulofilamentous material in combination with cytoplasmic bodies with a dense center, a dense core, and a clear halo. And this coarse material is also typically observed around the nuclei. And these features are exclusively seen, are very specific of this condition. Well, let's now move on and let's go to protein aggregate myopathies with vacuoles. And just to remind you where we are here. So these are protein aggregate myopathies that usually manifest later in life, and usually the protein aggregates appear in combination with rim vacuoles. And the first group of disorders I will go through is myofibrillar myopathies. While these conditions are familiar or sporadic muscle disorders that are defined morphologically by focal dissolution of the myofibrils, accumulation of the products of myofibril degradation, and ectopic expression of multiple proteins. This term indeed was proposed more than 30 years ago in an attempt to group under the same umbrella disorders that were described many years before by different authors and described using different terms. One important feature that unified this condition is that muscle fibers contain desmin aggregates. And because of this, these disorders were also named desmin storage myopathy or desmin-related myopathies. However, we know we later discovered that in addition to desmin, muscle fibers in myofibrillar myopathies contain a large number of proteins, and this renders these diseases as the paradigm of protein aggregate disorders of skeletal and sometimes cardiac muscle. Well, myofibrillar myopathy are very genetic, heterogeneous conditions from the genetic point of view. There are more than 10 genes that cause myofibrillar myopathy. These are desmin, alpha-B-crystalline, myotilin, ZASP, filamin C, BAC3, FHL1, some mutations in titin causing HMERV, DNA GB6, plectin, and some others that we will see in the next slide. Most of the majority of these proteins are sarcomeric proteins, and they all obviously express in a skeletal, sometimes also in cardiac muscle, and some of these proteins also express in other tissues, and this can explain some of the phenotypical features that are seen in some particular forms of myofibrillar myopathy. And for instance, the presence of lens opacities in alpha-B-crystalline is due to the fact that this protein is expressed in lens. And in addition to myofibrillar myopathy, mutations in these genes may cause other myopathy phenotypes or cardiomyopathy alone. These are some other genes that may cause myofibrillar myopathies. Well, I will not, of course, describe each subtype of myofibrillar myopathy, but I will summarize a couple of them. And the first one is desminopathy. Desminopathy is probably the best known and the best studied type of myofibrillar myopathy, and is the first myofibrillar myopathy that was molecularly resolved. The disease is caused by mutations in desmin gene on chromosome 2. It's a disorder that is transmitted following an autosomal dominant trait, but not infrequently patients carry de novo mutations, where autosomal recessive mutations are very rare and much more severe. Typical desminopathy patients present in young adults with a mean age at onset 32 years. The first symptoms usually consist of distal lower limb weakness, but some patients manifest with proximal weakness from the beginning or with a scapuloperoneal muscle weakness distribution, or they can initially manifest with cardiopathy or inclusion, or maybe can manifest with isolated respiratory insufficiency. As the disease progresses, other muscles are involved, and patients become wheelchair dependent some years after the onset of the disease. While cardiomyopathy is a very frequent complication of desminopathy, and the most it's seen in up to 75% of the patients. And very frequently, patients present with conduction blocks, and this is because of the fact that desmin is very abundant in the cardiac conduction system and Purkinje fibers. Some other patients present with with other dream disturbances, such as tachycardia. And as the disease progresses, the majority of patients develop hypertrophic, dilated, or restrictive cardiomyopathy. Here we have two patients showing this prominent weakness and atrophy of distal muscles. And this is a patient that was presented at the age of 14 years with a severe cardiomyopathy. He received a heart transplantation, but he later developed this skeletal muscle weakness that progressed and caused a respiratory insufficiency and died at the age of 33 years. So desminopathy is a very severe condition. From a pathology point of view, desminopathy typically show these patches of amorphous material that is best visualized on modified trichome stain. These areas occupied by this material or by these inclusions typically is devoid of oxidative enzyme activity and gives this grouped out appearance of the muscle fibers. And these inclusions correspond to protein aggregates that contain desmin, but many other proteins, including alpha-V-crystalline, filament C, dystrophin, myotylin, and many others. And under the electron microscopy, the ultrastructural hallmark is the presence of this typical granulofilamentous material that emanates at the level of the Z line. Let's move to myotylinopathy. Myotylinopathy is a disease or is a condition that has many features that make this disorder completely different to desminopathy. This is late onset disease. Patients manifest around the age of 53 years in our series. The disease is transmitted follow an autosomal dominant trait, but there are some sporadic cases. And the most common initial symptom consists of distal weakness on the lower limbs, but some patients may manifest with limb weakness, or a mixed combination of proximal and distal weakness. In contrast to desminopathy, cardiomyopathy and respiratory insufficiency is very infrequent in myotylinopathy. And here we have two patients. This is an 82 years old woman that was still ambulant, able to walk without support, which is completely different to desminopathy. And there are also important differences regarding the muscle pathology. Muscle biopsies in myotendinopathy show these more prominent inclusions, sometimes in form of spheroid bodies that show a strong congophilia. It's very often, or it's very common to find rim vacuoles, and also some muscle fiber necrosis with phagocytosis. And this leads sometimes to confusion or misdiagnosis with inclusion body myositis, especially at the beginning when the disease was described. While the areas occupied by the inclusions are devoid of oxidative enzyme activity, but these are irregular areas that differ from the drooped out fibers that we saw in desminopathy. And when we perform immunohistochemical analysis, there are strong or prominent protein aggregates containing desmin, myothelin, cyphilamine, etc. Here you can see collections of spheroid bodies very nicely decorated with the anti-myothelin antibody. Regarding the ultrastructure in myotendinopathy, the first or the initial lesions also began at the level of the Z line. There is disaggregation of the myofibrils, and there are accumulations of bundles of filaments of different electron intensity. These masses form these rounded or ovoid inclusions, sometimes or very often surrounded by groups of mitochondria. And in more advanced lesions or in fibers more destroyed, we can find accumulations of tubular filaments next to autophagic vacuoles. Well, let's turn to SASP myopathy or SASPopathy, and I would say that from a morphological point of view, I'm unable to distinguish SASP myopathy from myotendinopathy, but I will like to show you this particular observation we made some years ago when we discovered that muscle fibers in SASPopathy contain intranuclear rods. Intranuclear rods is a very rare finding that it's almost only found in intranuclear rod myopathy caused by mutations in ACTA1. But we observed the presence of intranuclear rods in one patient that was carrying a SAS mutation. And then we would decide to look at all cases with SAS mutations. We observed, or we had collected 7 SASPopathy patients, and all of them carried these intranuclear rods that were already seen on optic microscope, but were more easily seen under the electron microscopy. And if we use alpha-actinin, which is the major protein component of the rods, and look at the samples under the confocal microscopy, and we stain the nuclei with TAPI, we can see very well the intranuclear rods. And we were able to identify these rods in 7 out of 7 patients carrying SAS mutations, but also in some patients carrying myothelin mutation, as you can see in these slides. Well, and finally, regarding myofibrillar myopathies, I would like to say that irrespective of the mutated gene, myofibrillar myopathy samples have or show markers of autophagy, as revealed with antibodies against LC3, which is a marker of autophagy. We can see here LC3 colocalizing with desmin in a patient carrying a desmin mutation, or LC3 colocalizing with myothelin in a patient carrying a mutation in myothelin. Okay, and here I would like to include this slide just to to point out that protein aggregates can also observe or are also found in muscle fibers containing target lesions. And this is the muscle biopsy of the gastrocnemius of a patient suffering from acute denervation and reinnervation. And the muscle biopsy showed these typical target lesions, some rim vacuoles, and this sample contained, or these target lesions were stained with antibodies against desmin, alpha-bicrystalline, and ubiquitin. So it's important to know this situation in order to make a misdiagnosis of myofibrillar myopathy. Okay, let's move to another subject, and I will now go through vacuolar myopathies. And as the name says, or as the name implies, these are disorders that are defined by the presence of vacuoles in muscle cells. And for practical purposes, again, I will divide these vacuolar myopathies in three groups: rim vacuolar myopathies, and these usually also present small protein aggregates or small inclusion bodies. The second group is autophagic vacuolar myopathies that includes primary lysosomal disorders and others, and finally other vacuolar myopathies. Well, myopathies with rim vacuoles with a small protein aggregates or inclusion bodies are listed here, and you see that the number of disorders that belong to the group of rim vacuolar myopathies is very large, is huge, and the muscle pathology is very, very similar, if not identical. So in these cases, the muscle biopsy will not help us to distinguish among different conditions. And in the majority of cases, of course, we will, well, this is something that we always need, but here we will need to know very well how is the clinical features, and also we will need the genetic diagnosis. So I will not go through all these disorders, of course. I will only describe some of them. And the first one I would like to describe is actininopathy, a disease that was described only a few years ago in 3 Spanish and 1 Swedish family, and that suffered from a distal myopathy. And I must say that even I am a co-author of this paper, I do not agree considering actininopathy as a form of muscular dystrophy because of the features found in the muscle biopsies. Anyway, this is a patient that was not, another Spanish patient that was not included in the original publication. This is a man with a distal myopathy involving the anterior compartment of the legs. He had very high CK levels, which is something unusual in rim vacuolar myopathies. The patient had no respiratory or cardiomyopathy involvement. And the muscle biopsy of the anterior tibialis show large numbers of fibers containing rim vacuoles. And in addition to the rim vacuoles, there were some inclusion bodies. Many fibers contain cord-like lesions, and the inclusion bodies were immunostained with antibodies against P62, ubiquitin, phosphorylated neurofilaments, TDP43, valosin, and many others. So I would like to point out here that the presence of these small aggregates, it's completely different to what we see in myofibrillar myopathies. And therefore, I would never consider these disorders as a form of myofibrillar myopathy. And the second aspect that we'd like to remark or to point out is that the presence of P62, ubiquitin, TDP43, etc., is not found only in inclusion body myositis. It's a finding that occurs in all the myopathies with rim vacuoles and inclusion bodies, as we have seen here. Well, when we look at the electron microscopy, alpha actininopathy shows these sarcomeric abnormalities with irregularities of the Z line, some fibers with more advanced lesions, so show a complete disorganization of the sarcomeres, and also collections of tubular filaments in combination or next to the to some autophagic vacuoles. Another condition, which is very similar from the morphological point of view, but very different from a clinical point of view, is VCP myopathy or inclusion body myopathy with Paget disease of bone and frontotemporal dementia. Muscle biopsy in this condition also shows trim vacuoles, small inclusion bodies that react with antibodies with palosin, ubiquitin, phosphorylating neurofilaments, but also against TDP43, P62, etc. And under the electron microscopy, that is typically the combination of autophagic vacuoles and tubular filaments, as we have seen also in actininopathy. Well, this is just to illustrate the presence of P62 and TDP43 and ubiquitin in inclusion bodies found in Valosin myopathy. And the last disorder I will show you belonging to this group is GNE myopathy, formerly named Nonaka myopathy. This is an autosomal recessive condition caused by mutations in GNE. As you know, GNE is involved in the biosynthesis pathway of sialic acids. Well, anyway, this disorder manifests also with a distal myopathy at the beginning with typical preservation of the quadriceps. And the muscle biopsy again shows large numbers of fibers containing rim vacuoles and some small inclusion bodies that are can be detected with antibodies against P62, ubiquitin, or any other proteins, as I have shown you in my previous slides. Well, regarding non-hereditary conditions, there is an acquired disorder that show have trim vacuoles in the muscle fibers, often in combination with inflammatory infiltrates. And this disorder is inclusion body myositis. You probably know very well is a disease that manifests in adults, usually older than 50 years, and typically involves the quadriceps muscle, the flexor, the finger flexors, and anterior tibialis, but later spreads to other groups. And around a third of patients present swallowing difficulties. And the muscle biopsy showed these endometrial inflammatory infiltrates, some of them partially inviting normal fibers. The infiltrates correspond basically to CD8 lymphocytes, and this occurs in combination with rim vacuoles and some inclusion bodies that are stained with ubiquitin and P62 again. Another typical feature of inclusion body myositis is the presence of mitochondrial abnormalities with large numbers of COX deficient or COX negative fibers and overexpression of the major histocompatibility complex class 1. Well, even the rim vacuolar myopathies I have described in these previous slides have features of autophagy. For a number of reasons, these conditions are usually classified separately from those disorders known as autophagic vacuolar myopathies. And autophagic vacuolar myopathies usually refer to primary lysosomal disorders, including Danon disease, X-MIA, and Pompe's disease, and non-primary lysosomal disorders, including some toxic myopathies. And one common aspect here of some of these conditions is that the vacuoles show sarcolemmal features, as we will see in the next slides. So I will start with primary lysosomal disorders. And the first one is Danon disease. Danon disease is an X-linked dominant disease caused by mutations in LAMP2, a gene that encodes a lysosomal associated protein 2. Well, the disease is clinically characterized by the triad of hypertrophic cardiomyopathy, intellectual disability, and skeletal myopathy that is usually mild. And the muscle biopsy typically show these granules within the cytoplasm of the muscle fibers. These granules correspond to small vacuoles, and these vacuoles show sarcolemmal features as revealed with antibodies against membrane proteins, including dystrophin, or as revealed with acetylcholinesterase activity. If we perform immunofluorescence, LAMP2 is completely absent in the samples of Danon disease patients, and also is absent if we perform Western blot. And under the electron microscopy, there are collections of autophagic vacuoles that are surrounded by a membrane. This corresponds to lysosomes. While X-MIA is a disorder, I will go through this slide quickly. It's almost identical to Danon disease from a morphological point of view, but in contrast to Danon disease, here LAMP2, the expression of LAMP2 is preserved because this disorder is caused by mutation in another gene, a gene that encodes an ATPase proton pump. Well, another a feature typical of this condition that is also seen can be also seen in Danon disease is the presence of major histocompatibility complex expression at the surface of muscle fibers, also complement deposition, and also calcium deposition in the muscle cells. Okay, and the third lysosomal, primary lysosomal disorder that I will show you is Pompe's disease. Pompe's disease, as you know, is glycogen storage disease type 2 that is caused by biallelic mutations in GAA, a gene encoding acid maltase, which is a lysosomal enzyme that breaks the glucogen into glucose within the lysosomes. And as a consequence, there is a progressive accumulation of glycogen in skeletal muscle, heart, and other tissues. You know very well that there is a correlation between the clinical phenotype and the residual enzyme activity levels from the classical form to late onset forms. And this is very well seen in the muscle biopsies. We almost never do muscle biopsies in Pompe's disease because we have very easy ways to make the diagnosis. Anyway, you should know that in the classical form there is massive presence of massive vacuolated fibers in the cytoplasm of muscle fibers, and these vacuoles contain pus, as revealed in pus staining, and this glycogen, or this glycogen accumulation disappears after the stage digestion, indicating that it really corresponds to glycogen. In the late onset Pompe's disease form, the muscle pathology is less severe, and usually what we found some are collection of multiple small vacuoles that also contain glycogen. These vacuoles display acid phosphatase activity because acid maltase, as I mentioned, is a lysosomal disorder. And well, the areas that are occupied by these vacuoles are devoid of ATPase and oxidative enzyme activity. And we can also observe markers of lysosome proliferation using antibodies against lysosomal associated protein membrane 2. And we can also find markers of autophagy as revealed with LC3 immunofluorescence. Under the electron microscopy, what is typical of Pompe's disease is the presence of membrane-bound glycogen accumulation. But in addition to this membrane-bound accumulation of glycogen, we can also find free glycogen. And you know that this free glycogen does not cannot be removed with the enzymatic replacement therapy. Well, and we go now to the non-primary lysosomal disorders, and I will briefly explain or show you colchicine toxicity. As you know, colchicine is a drug that is used for the treatment for gout and other conditions. Colchicine binds to tubulin and impairs lysosomal transport along the microtubules, and therefore it inhibits lysosomal exocytosis and blocks autophagy flux, autophagosome flux. And muscle biopsies in patients affected with colchicine toxicity show small, very small, and punctate vacuoles in combination with some other larger vacuoles that, when observed under the electron microscopy, correspond again to lysosomes or autophagic vacuoles surrounded by a membrane, in addition to some sarcomeric disorganization. And in this pathology, or this myopathy, also shows sarcolemmal features, as revealed with dystrophin antibodies, show markers of autophagy when we use LC3 antibodies, and typically show expression of alpha-bicrystalline and overexpression of the major histocompatibility complex class 1. Well, I now realize I forgot to mention, and I think it's important that I mention that in Danon disease and also in colchicine toxicity, there are abnormalities at the level of the basal lamina of the muscle fibers, and there are formation of loops and redundancy of the muscle lamina. And this is probably what explains, or the reason why these conditions show overexpression of the major histocompatibility complex class 1. I will briefly mention this disorder that I think it's new member of autophagic vacuolar myopathies, non-lysosomal primary in origin, and this is PLIN4 myopathy. This is a condition caused by an expansion in PLIN4. PLIN4 encodes perilipin 4, which is a protein that codes the phospholipid monolayer that surrounds lipid droplets. This gene, PLIN4, contains 28 short tandem repeat motifs of 99 base pairs. Well, so very recently, an expansion in this gene, in this repeat motif, was identified in a family, in an Italian family, that remained without a diagnosis for many years. Even there, it was known that there was a linkage to a locus on chromosome 19. Well, in this family, suffering from a distal myopathy, the tandem repeat motif was of 31 instead of 28 tandem repeats. And from a pathological point of view, this disorder is very peculiar because it shows the presence of large numbers of rim vacuoles that are seen within the cytoplasm of the muscle cells, but also under the sarcolemma, or at the level of the sarcolemma. And this disease typically shows this sarcolemma labeling when you use P62 and also perilipin, which is perilipin 4, which is the mutated protein. Under the electron microscopy, there are large numbers of autophagic vacuoles and also some abnormalities at the level of the basal lamina with duplications or redundancy of the basal lamina. Just to end up, I will briefly mention two other vacuolar myopathies. The first one is McArdle's disease. Again, we almost never perform biopsies in these patients, but anyway, just to know how the muscles of these patients look like. Well, they show subcircumflemal vacuoles that contain glycogen as revealed with PAS staining. This glycogen is observed free, it's not surrounded by a membrane, and the demonstration of the lack of phosphorylase activity allow us to establish the diagnosis. And finally, glycogen storage disease type 3 is another glycogenosis that shows large numbers of vacuoles in both types of fibers, and these vacuoles contain accumulation of glycogen that corresponds to free glycogen that is found under the sarcolemma, but also between the myofibrils. Okay, so that's all. I think, I hope you are still there and I have not troubled you with so much information. This is my group, and this is a nice view of the old buildings of the hospital where I work. Thank you very much for your attention.
Thank you very much, Montse. Very interesting and picturesque, I would say, presentation, as always when we are talking about pathology. I'm going to give you a prize because I've just launched a poll so that the members that are attending this presentation may answer if they think they still need muscle biopsies. So we'll leave people some time to answer. This is anonymous, don't be afraid, even if you say no. But we'll see in a minute what's the answer that people think is most adequate. We have 19 votes, so few to go. $21. Okay. Okay, so I'm going to end the poll and I'm going to show the results. So as you see, yes is 95% and no is just one attendance, so 5%. So we were able to keep your job. I should congratulate you on that as well. And regarding questions, as usual, people start shy, and then when we don't have any time, they start asking things, but it's my turn as usual to start the question and answer period. And I would like to basically discuss with you and ask you about the new extended phenotype that we call multi-omics, as you know. And I think that probably one thing that I noticed during your presentation is that you included MRI images, which makes sense in many of the pathologies, and they give you support for the diagnosis besides the muscle biopsy. And I was wondering if you are aware of some kind of morphological combination of modalities between pathology and MRI imaging, for instance, or other, for increasing the accuracy and the yield of the diagnosis.
Well, thank you for the question and for the comments. I think someone, and I don't remember who, but maybe it's I don't remember his name. There is a guy working in Rome that was doing some work to make a kind of correlation. Matteo Garibaldi, I think. Matteo Garibaldi was trying to do some studies to make a correlation between the MRI images and the muscle morphology, yes.
Mm-hmm.
But so far, there is no publication regarding this, as far as I know.
Okay. And what about artificial intelligence and machine learning algorithms, either adding or replacing? I don't know.
Well, this is something.
The clinicians.
This is the current fashion to use intelligent artificial intelligence, and even in my group, some people who do not work with muscle morphology are trying to encourage me to develop a kind of program. And I'm not saying it cannot be useful, but I'm not an enthusiastic person of this kind of tools. I know, for instance, in the Institute of Myology, Norma and I don't remember the name of the guy who
Bruno
Badot, yes, they have collected a huge number of very nice and very well processed muscle biopsies, and they were trying to develop a program. I think you need to have a very perfect slides, very well processed, and then try to find some kind of algorithms. This can be useful for some pathologies, but probably not for all. Anyway, I know that this is something that it's going to appear. Let's see how it helps.
Yes, fingers crossed that you keep your job. Jonathan, do you want to unmute yourself and put your question loud?
Hi, yes, of course. Great talk, thanks. Well, I'm still in training, but I wondered how do I start learning all these pathological features and patterns? Is it purely exposure, or are there any good books or reference works you can recommend?
To learn pathology. Jonathan is asking, what do you recommend to learn pathology in terms of reference books and other sources, and how do you see the best method for learning pathology?
Well, there is a Bible about muscle biopsy, and it's the book of Dubovitz. There is a relatively new edition of the book. The book is Muscle Biopsy, a Practical Approach, and is edited by Victor Dubovitz, Anders Oldfors, and Caroline Seury. You can find it, you can buy it on online, or you can have a physical book, and it's very good.
Okay, thanks.
Very well. But I believe that what you haven't said there is that you still have to go to a good lab and to a good center or several.
Yes, of course.
And learn by example and observation.
Yes, and you need to examine lots of samples before you are ready to make or to become an expert in muscle pathology, of course.
Yeah, so I think we have had a very pleasant hour discussing this theme, and I think people went from here with more ideas. There were already questions about if this presentation would be available. Yes, it was recorded, and we'll make it available in our website, on the webpage where we talk about the webinars, and also on the YouTube channel of EURO-NMD. And thank you very much, Montse, and It was a pleasure to have you, and it will be a pleasure to have you again very soon, I'm sure. So
Okay.
thank you to all that have been present, and goodbye until next time.
Thank you very much.